we're going to continue with the modeling signal propagation and diffusion section, still in chapter 3, and hopefully we'll finish it today. So, sorry, about the model of the signal propagation, the first thing we get is the intriguing analogy between the radio waves with the electromagnetic signals and the calcium waves we have been discussing. There's similarities, and we'll be able to benefit from similar ideas and carry these ideas to the field of calcium signaling. The second thing is the analysis used in understanding the operation of the calcium waves in the environment. Sorry. And also other biological signaling mechanisms. These will also include the oscillatory pattern for example, used in the pheromone information transport. And finally, there are the direct implementations of the calcium wave that have been already proposed in 13 by Suda and Moore. The signal propagation and diffusion are the basic components of the nanoscale communication mechanisms. So if you look at the fundamental nature of how these signals of molecules, signals implemented with the transmission of molecules, is spread among a collection of cells for communication in between, we should first keep in mind that the transmitter cell is actually secreting some molecules into the environment, creating a higher concentration of those molecules in the environment, and these molecules will typically be spread until they're received at the receiver, and then the receiver will try to understand what the transmitted signal was. So let's try to define and derive the characteristics of this communication and look at the characteristic distance of communication. So we should first understand how diffusion is actually working. Remember, we discussed about the Brownian motion, but let's look at it in more detail and try to formalize it. So diffusion is actually a time-dependent spread of the molecules in the environment. So there's a mass transport of the signaling molecules initiated from a spherical source for the sake of the example. So according to the equation here, the C here is the concentration of the messenger molecules in the environment, and D is the diffusion coefficient of the medium, the extracellular medium. So this will be dependent on R, and we can formalize it this way. Here the diffusion equation is, this equation is applicable because it describes the movement of the particles in a random manner. Rather than looking at the collisions each molecule is experiencing, we're looking at the flow actually. How this high concentration of molecules close to the transmitter cell diffuse in the environment in time. Of course, depending on the distance. And we're not looking into the Newtonian physics, but we're looking at diffusion. So the boundary conditions for this transmission can be represented as in here, where F0 is the signaling molecule production rate, and this is what we have at the distance of rho, which is actually the cell radius. So this is one of the bounds, and the other extreme is where the distance is infinity. In that case, the concentration will of course diminish down to 0. We will not go into the derivation of this stuff. Please refer to your textbook and read section 3.4.1 for the derivation, but we will not go into that derivation during the lecture. Now, an interesting result we obtained from here is, from this analysis, is that typically you would expect that if the diffusion gets faster, the molecules will diffuse towards the destination in a faster manner. So it will work better for longer distances. However, it actually works the other way around. Since a slower diffusion rate typically allows a higher concentration to build up, it will not immediately diffuse in all directions. Before the random movement dilutes the density of the molecules. So it's important to note that this analysis assumes the ideal case of cell-secreting molecules from a central location, where the central location is the transmitter cell itself. Now, cytoplasmic, sorry, I have a problem with my throat today. The cytoplasmic calcium concentration now can be increased either with the release of intracellular stores. Remember, we briefly discussed this, like the endoplasmic reticulum is serving as a calcium store, or via the influx into the cell. There are already calcium ions in the environment. These will also diffuse into the cell or pumped into the cell with the calcium pumps. So while the calcium signaling is a typical signaling mechanism, it is also affected and controlled through other signaling mechanisms, typically with secondary messengers. So there are secondary messengers that constitute actually what is being propagated. But these messenger molecules have a side effect on the calcium concentration inside the cell, so that they trigger the release and later on getting back to the calcium pools of these calcium ions, so that you can control the calcium ion concentration in the cell. Typically, we will have a small number of these messenger molecules that will trigger high concentrations of released calcium from the internal stores. And these, for these secondary messenger molecules, there are a limited number of different types of secondary messenger molecules, but the mostly known one is the inositol triphosphate, which is shortened as INSP3, or a shorter form of this, I will generally use that term, would be IP3 molecule. So we'll be focusing on that. But just know that there are also alternative secondary messenger molecules. The calcium release from the cell is relatively quick when required, and it fully deactivates within a few tens of seconds. The release is due to the clearance of the calcium from the cytosol by resequestration into other organelles, collection into the other organelles, typically endoplasmic reticulum and the mitochondria. These are serving as the calcium pools. As well as you get rid of these calcium ions inside the cell by pumping them, for example, out of the cell, using these pumps, calcium and sodium exchangers, and also the calcium ATPases. If you look at the calcium ion intake from the environment inside the cell, the concentration gradient for calcium across the cell membrane of resting cells, that means without the first trigger, differs by approximately 10,000 times. So there is much difference. So there's much larger concentration ready in the environment to go into the cell. In addition, the cell membrane has a hyperpolarized resting membrane potential. And this hyperpolarization actually means the cell's membrane has a negative charge, but remember, calcium ion is positively charged. So typically this will attract the calcium ions towards. And the combination of the concentration difference between the extracellular environment and the cytosol inside the cell, and also the charge difference between the ion and the cell membrane, they both serve to attract calcium ions inside the cell. So even with a low membrane permeability for calcium ions, a small increase in permeability results in a large calcium influx. This is due to this huge difference in the concentration for extracellular environment, between the extracellular environment and the cytosol. So the permeability increase occurs when we open the ion channels on the cell membrane, and then the calcium ions can enter the cells through these calcium ion channels and increase the concentration inside. These channels, there are various different types of channels. One is voltage operated channels. Another one is the secondary messenger operated channel. We'll be talking about these. In the case of voltage operated channels, for example, by changing the voltage on the cell membrane, you open or close the channels. Remember, we had the video about this earlier. With the case of secondary messenger operated channels, the arrival of secondary messenger molecules typically cause these channels to open so that the calcium ions may flow into the cell. Store operated channels are, we have these calcium stores or the pools. So by playing with the calcium pool receptors, we can start the release of calcium ions. And also receptor operated channels, with the arrival of an agonist to the receptor, you trigger the release of calcium ions. So these in more detail. If you look at the voltage operated channels, these are activated, as I said, with the change in the polarity of the cell membrane. And second messenger operated channels are activated by any of a given or a small number of secondary messenger molecules. As we said, IP3 is the most well-known one. But in addition to IP3 and other inositol phosphates, there are also cyclic nucleotides that are used for a similar purpose. And for the case of the store-operated channels, these are activated by depletion of the intracellular calcium stores, and the receptor-operated channels are activated by the direct binding of a neurotransmitter or a hormone agonist. The specialized transmembrane proteins typically carry the ion currents in and out of the cell. So these proteins are actually serving as the ion channels. At rest, these channels are typically closed and they're impermeable, which means they will not let the calcium ions leak through the channel. However, when you cause these channels to open, for example with the voltage, either by changes in the voltage in the membrane potential or by some certain ligands that will trigger the release of, we'll see the figure about this. A ligand will, for example, stick to the channel, and this will cause the channel to open. When this happens, the calcium will flow from higher concentration, which is true for the extracellular environment, to the lower concentration, which is the cytosol. There are various types of ion channels, as we have seen, and they may be channel length, or this is also called ligand-gated channels, LGC, enzyme linked, or G protein coupled. That's what we're going to discuss in more detail. Intracellular or voltage gated. Ligand-gated channels typically mediate fast synaptic transmission at the synaptic clefts, at the points where the neurons, for example, touch each other. So this is the synaptic cleft. So this is the axon of the presynaptic neuron, and this is the other one. So this is where the get close to each other, the synaptic region. The nerve impulse typically triggers the operation here. That will allow the calcium concentration in this cell to increase. What happens is this axon or the cell here takes in calcium from the extracellular environment. The synaptic vesicles typically contain the neurotransmitter molecules, which are going to be used here. These vesicles will be carried to the synaptic cleft here, and they will diffuse in a short distance towards the ion channels of the other cell. These green neurotransmitter molecules, some of them with diffusion will arrive at, as you see here, it will arrive at the ion channel, and it will stick on the ion channel. And when that happens, when it binds to the channel, this one will open. Here you see a closed one. Some are open, some are closed. Okay? So this way, this neuron here triggers a change on the ion channels of the other neuron. And these channels, when they open, they let the calcium flow into the cell so that the calcium level inside the second cell increases. Okay? This way, actually, you're not sending the calcium ions from one cell to the other, but you're doing something on the other cell that will affect the calcium ion concentration of the other cell. This way you create the channel. Here we see this ion channel in more detail. When this neurotransmitter diffuses from the first neuron to the second, the neurotransmitter will bind to the receptor here, and that will cause the channel here, which is on the postsynaptic membrane of the second cell, to open. And later on, this neurotransmitter will depart from the receptors here, and it will also be part of and it will be degraded so that it will not affect another channel. It will not bind to another channel. Its duty is completed, so it will just decompose. This way, a nerve impulse will travel from one cell to the other. Inside the neuron, this will be transmitted as an electrical signal, but at the synaptic cleft, we have a chemical communication between the neurons, which increases the calcium ion concentration in the second neuron, which will trigger another electrical signal to be generated inside this neuron, and that will be propagated as an electrical signal to the next synaptic cleft. An agonist, as you will remember, is a special type of molecule that binds to the receptor of a cell, and it causes a change in that receptor. It causes a change that will affect somehow the cell. Okay. Besides agonist, we had one more thing. That was what? Do you remember it? Antagonist. And what was the difference between agonist and antagonist?
Antagonist was stick to the molecule so that agonist cannot.
Right. Antagonist typically blocks. That also attaches. But the difference is antagonist does not affect the inner mechanisms of the cell, so it does not trigger any chain reactions inside the cell. What it does is it will block the receptor so that the agonist may not attach to the receptor. Okay? So the agonist's efficacy is a property that distinguishes it from the antagonist, which is what we just discussed, which binds to a receptor but does not cause any chain reaction inside that cell. An agonist may have a positive efficacy, causing an increase in the receptor's activity, or it may also have negative efficacy. In that case, it will cause a decrease in the receptor's activity. So there are different types of agonists. Under some conditions, calcium can enter cells via the sodium calcium exchangers, which are actually operating as pumps, operating in the reverse mode from lower concentration, for example, to higher, if necessary. Excitable cells are available in most animal organisms. For example, we see the excitable cells in the nerves and also in the muscle cells. These contain voltage-gated calcium channels, as we discussed, which means the passage of the calcium ions is controlled by the voltage or the polarity of the cell membrane. But we don't have such voltage-gated calcium channels in cells that are not excitable. Receptor-operated channels typically open upon the binding of an external ligand, typically a neurotransmitter, as we have just discussed. And these are also available on the excitable cells. Second, messenger-operated channels are found less often, and they seem evenly distributed between excitable and non-excitable cells. And the store-operated calcium permeable channels appear to be widespread, present in all eukaryotes, from yeast to human. You know what a eukaryote is, right?
A cell
with a nucleus. With a nucleus membrane, therefore the genetic material is enclosed inside the nucleus. In prokaryotes, the genetic material is spread throughout the cell. There's no nucleus. The concept of using calcium waves, or the existence of calcium waves with store-operated calcium ions, was proposed in 1986. The idea is originated from a series of experiments that were done with the cells while investigating the relationship between the calcium ion release and the internal stores, the endoplasmic reticulum and mitochondria. Calcium entry into the cell through the channels, and refilling of the calcium store, that means storing the released calcium back into those pools. It appeared that the amount of calcium ion in the stores controlled the extent of calcium ion influx from the environment inside the cell in non-excitable cells, which was originally called a capacitative calcium entry. And when the stores were full, the calcium influx did not occur, but as the store is emptied, then calcium ion intake is increased. There are different types of cells that make use of intercellular calcium waves. So there, for example, the hepatocytes, like the cells in the liver, pituitary cells, the cells that are used in the blood vessels, and also the tissue cells, all these make use of calcium ion waves. Also, the newly fertilized eggs have demonstrated repetitive spikes in the concentration of the intracellular calcium concentration. This is typically either caused by the hormone or the neurotransmitter signaling mechanisms. Note that for the calcium ions, the calcium ion waves may occur in two different ways. One is with the spikes, so there's a sudden increase and then a decrease, and that decrease is pretty slow. And the other is you start the wave and it shows a sinusoidal pattern. Probably the second method, in which you have the sinusoidal transmission, is the one that we could exploit more and modulate to transmit information through. The period of these oscillatory spikes we mentioned in the concentration range from less than 1 second up to 30 minutes. And once you have a spike, after that the cell has to wait for some time, so before a new spike can be generated. So this actually limits the data rate you can achieve. So it has been clear since the discovery of these oscillations that they encode and transmit information between the cells naturally. We will follow the model given in reference 17 where we will assume that the internal agent triggers the production of IP3. This will cause the cell to release calcium ions that were previously stored in the calcium ion pools in, for example, the endoplasmic reticulum. This will cause the increase of the calcium ion cytosolic calcium ion concentration. And also remember that the cell can easily replenish calcium ion into its internal stores by pumping in more calcium ions into the cell if required. The operation of the mechanism is as follows. Remember the agonist that was binding to the receptor. So when that agonist binds, it actually starts a sequence of chemical operations that are related to the receptor, the G protein, the PIC, and these are used to actually hydrolyze the the inositol lipid phosphatidylenosyl, biphosphate into DG and IP3. The moral of the story is the following. An external agonist will arrive at the receptor. It will bind with the receptor, which will affect the G protein, which will cause another reaction for the PIC to hydrolyze into DG and IP3. For our case, the important thing is not DG, but it is IP3, because the IP3 concentration actually triggers, the IP3 molecules trigger the inner calcium pools. Okay? So with the arrival of the agonist, the receptor has triggered the G protein, which caused the PIC to produce DG and IP3. And this hydrolysis of the PIC into DG and IP3 caused actually the hydrolysis of the water, which will be used to break down the polymers. So this IP3 released from here then controls and modulates the release of the calcium ions from these inner pools. IP3 sensitive stores inside the cell, and this will increase the cytosolic calcium ion concentration, as you mentioned. So the rate at which this occurs, let's denote it by V1, and we will be discussing actually this operation here. So this operation, remember, affects PIC and GP, and this causes IP3 and DG to be released. So let the rate of this be V1. I'll talk about beta later on. Okay? Let this be at the rate of V1, the release of IP3 ions. It's also this also indirectly controls the influx of external calcium ions from the extracellular environment into the cell when necessary, using that capacitive mechanism we just discussed in the previous slide. So each IP3 molecule actually controls the flow of calcium ions from these inner pools with a constant rate, with a constant flow. Okay, let that be controlled by beta. So for each IP3, you can take this. For each IP3, I have beta calcium ions, for example, released into the environment. But I have the arrival rate of IP3 molecules denoted by V1. So for each one beta calcium ions, so in total it would be V1 times beta. Therefore, in this figure, with the arrival of the agonist at the receptor, at the rate of V1, IP3 molecules will be released, but each one triggering at a rate of beta, the overall rate of calcium that would be created due to this operation would be V1 times beta. In addition, let Y be the concentration of the IP3 insensitive pool of calcium ions. So here we have the calcium ion pools. Let Y be the concentration here, okay? And let Z be the concentration of calcium ions in the cytosol overall, okay? So that's the important thing for me actually. I want to see how Z changes according to time, okay? So I want to find the effect of all these on Z. I already have a concentration of Y already stored here, and IP3 is, remember, released. So the concentration level of calcium ion will be typically low in the beginning, so that means Z will be small in the beginning. But with this triggering operation, I will be able to increase the value of Z. So at the rate of V2, calcium ions will be released from the IP3 pool. So, sorry, they will be stored into the pool. So let me explain everything actually from the, over the figure. This is our pool, okay? The arrival of the agonist has triggered the release of IP3 molecules. The IP3 molecules here will trigger the calcium pool, and they will, the calcium pool will release calcium ions at a rate of V3. Meanwhile, calcium ions are also flowing into the pool. So while you're throwing some out, some also flow in at a rate of V2. Okay? Some also will diffuse due to the concentration difference. Some of them will also diffuse without the effect of IP3. So let that be denoted by K sub F. Okay? This is what's going on with the calcium pool here. But if you look at the overall cell, the small calcium ions will still diffuse into the cell with a rate of V0, but also diffuse out of the cell with a rate of K. Okay? When you consider all of these flows, you can follow what will happen to DZ. What will happen to, Z. The calcium ion concentration in the cytosol, which is what I want to follow. Okay? And that would be actually, we'll see it in the next slide, Z would have plus and minus terms. Plus are the ones that cause the increase in Z, and minus are those that cause a decrease. So V1 times beta will be positive, that causes an increase. V3 will also cause an increase in Z. This V0 will also cause an increase in Z, whereas K will be negative. V2 will also be negative because it will cause Z to decrease. It's because this is storing calcium ions in the pool. This Kf is also positive for Z. Okay? However, some of these are dependent on the concentration, some are not, as you'll see. So let that V0, which was flowing from outside the cell into the cell, let V0 be the rate of influx into the cell. K, as we said, is the efflux out of the cell into the extracellular environment. Kf is the rate of passive leak of calcium ions from the IP3 sensitive pools. And as we said, V1 times beta is used to describe the influx of calcium ions into the cytosol. And we're going to show the rate of Z in the following slide, as we said. We can see that it is the sum of the input rates minus the output rates. So V0 is the input rate. Let me explain it here. V0 is going in. Does it depend on Z? No, because the main factor here is the extracellular environment, which is huge compared to the cell. So it is, the concentration will not change anything. So we have plus V0. Okay? Plus V0. Okay? This was, remember, due to the IP3 molecules. IP3 molecules were generated with a rate of V1, and each one has an effect of beta. That's why it's V1 minus times beta. V3, as you will remember, is the release from the calcium pool. So we have that V3 there. Now, V2 was the diffusion, the leakage into the cytoplasm. So that's why it's negative with respect to Z. However, if you look at this Kf, which is leakage, this one depends on the concentration here. The higher Y is, the more calcium ions will leak. So it is Kf times Y. Okay? That's what we have here. And also, sorry, if you look at the leakage from the cell to the extracellular environment, now the concentration here is important. The higher this concentration is, just similar to what we had here with Kf, the higher the cytosolic calcium concentration is, the more leakage we will have. Therefore, that one's also K times Z. Okay? And that's what we have here. Once again, the change in Z with respect to time is dependent on arrival or diffusion from the extracellular environment into the cytosol and the calcium ion release due to IP3 release and calcium ion release from the internal calcium pools and also calcium ion release that is due to leakage, not triggered by IP3, but that's due to leakage, minus leakage outside the cell from the cytosol, minus leakage into the calcium ion pools. If you look at what is going on inside this cytosolic, inside this calcium pool, sorry, it's similar thing. We have V2 causing an increase in Y. Remember, Y is the concentration of calcium inside the calcium pool. So we have V2 as positive, Kf and V3 as negative. So we have V2 positive, Kf times Y, and V3 as negative terms here. That is the rate of change in the calcium ion concentration inside the calcium pool. So to summarize, the signal begins when an external signal triggers a sequence of chemical reactions that will release, the ultimate product is the IP3, And this IP3 further triggers release of calcium ions from the calcium pool into the cytosol. And this increase in the intercellular calcium concentration is creates a positive feedback mechanism which increases V3, the release of more calcium ions.